sorry, there was a little technical glitch there. We're going. So what should you do this week? We started talking about Chapter 7, and we're going to get through almost all of Chapter 7 today, and then on to Chapter 8. Then we finished that plowing assignment, and so people handed it in or sent it to me, and now we're doing that Northwest assignment. And I heard that a student stopped by my office yesterday when another student had left a bag of stuff inside my office. So apologize about that. There's that bag of materials outside my office now to complete that Northwest drawing assignment. And then you're working on your term papers, and I've talked to everybody. I think people are making good progress on that. We just are going to roll forward. So we're talking about chapter, and we got to introduce kind of a little review of these sort of three components of what the stories fit, and that is the stories include the cytoskeleton inside the cell, inside of every animal cell, and in the cytoplasm of that cell. Then in the cell membrane, the plasma membrane, there are these cell adhesion molecules, proteins that we've learned a little bit about. They represent them so far as macaroni. And then this extracellular matrix, or ECM, that is the material that's left after an organ is decellularized, so it has lots of information. And so one of the first key things that we introduced already from that example is that this extracellular matrix is, maybe if you've heard of things like collagen, then you would be aware that the extracellular matrix has a structural component, a strength component, mechanical component, and with that decellularization we see, in addition, and really significantly for us, the extracellular matrix also has information embedded in it, different molecules in different places at different concentrations, and that's how cells know where to go when they're migrating. And it's one of the big factors of how they know where to go when they're migrating, and making connections. And so we're just going to kind of turn to each of those categories and introduce them, like what are the terms and molecules that we need to know about and what's their basic role. And so here's the first one, the cytoskeleton, and this is stuff that you would have learned about in intro-bio, and then we're going to put it into some examples. So just to introduce those, the three main components of the cytoskeleton are all polymers of proteins, and they can be assembled and disassembled, so they can be polymerized or depolymerized, and they fall into these categories. Most people, when they talk about the first one, they just say microfilaments. I like to always think actin microfilaments, because it helps me tell the difference between microfilaments and the next one, microtubules, because they play different roles. And when we talk about microfilaments or actin microfilaments, they're involved in changes in cell shape. And we've already seen examples that we'll look at that involve changes in cell shape. When we think of microtubules, we think about stuff moving around inside of a cell. Okay, so it's really important to distinguish those roles. And there's some overlap of those roles in real life, but they really fall into those big categories like that. And then the third one, they're called intermediate filaments, and intermediate filaments play really important roles, typically sort of structural roles, especially structural roles. Like there are intermediate filaments inside the nucleus that help give it its kind of round shape. The intermediate filaments they don't change very much. They kind of get built and they're pretty stable. We know that in a lot of species, when the cell's going to divide, the nucleus disappears. And it disappears because the intermediate filaments are depolymerized during mitosis. So you can change the intermediate filaments around, of course, but they're just a little bit more stable and thought of more stable and constant structures in a cell. And as a result, we would say a developmental biologist thinks intermediate filaments are pretty boring. And the stories are in the actin microfilaments and the microtubules. And so we want to tell some stories about that. And just to put them in context, so here's a cross-section of an antivertebrate embryo when the neural tube is folding. And you can see it's folding up, it's pinching off, and those changes are changes in cell shape. So an example of the role of actin microfilaments would be, anytime cells are changing shape. And the book shows that, talks about that, and we'll show examples like this type of example here, where just by having these actin microfilaments positioned in certain locations, we can change the cell shape. And that's what's happening when the neural crest cells fold out. If you look at individual cells, the individual cells are changing shape. Some are getting narrower on one surface, some are getting narrower on the opposite surface. And you can get folds made like that. Another example that comes up is the blastopore. When the blastopore forms, that's that dent that forms. That's going to be the eventual anus and deuterostomes. And when that blastopore forms, it's really just cells, individual cells, changing shape. And they're changing it in different shapes to get the different curves to have them happen, like that. I want to just point out the neural crest cells because that's where they start. And we're going to do an assignment on them, and they always start along the dorsal midline in all vertebrates. They're going to crawl to a variety of different locations. We're not talking about crawling right now. We're just talking about cells really staying in the same place and changing shape. And the way the shape change is happening is that we call these actin microfilaments because they are made of polymers of actin. And so this is what an actin microfilament looks like. And it's just a long polymer of actin. And when you look under a microscope at the organization of actin microfilaments in a cell, they look like somebody has just taken some straw, uncooked spaghetti pieces and just kind of throwing it out on the table. They look like they're just scattered around, but they do have some organization to them, and they can change that organization around really fast, and they do it by depolymerizing the actin microfilaments and building them somewhere else, polymerize them, and that can happen. That happens really fast, and the way that the actin microfilaments are depolymerized is that the actin is removed from one end of it. So they grow from one end and they depolymerize from that same end. Okay? And so they're involved in changes in cell shape. How do those changes take place? And the answer is there is another protein that's bound to them and can slide the microfilaments past each other. And that protein is myosin. And if you learned about some physiology, you'd probably recognize that muscle fibers in our arms and legs and so forth, those muscle fibers have myosin motors, and the myosin motors use ATP to pull the actin microfilaments past each other. So we have actin and myosin sliding past each other, and the cytoskeleton is built on that same strategy. It's not as organized. I started off by saying that microfilaments are scattered like scattered spaghetti. A muscle is like when the spaghetti's in the package and all the actin microfilaments are all lined up in parallel, and you get a lot of directional force. So it's the same story in the actin microfilament cytoskeleton as it is in the muscle. The purpose of the actin microskeleton is to change shape, to contract and lengthen. And the neural crest cell migration is a really good example of cells moving because they're changing shape. So that's a really good example that dives into that. The actin in our bodies is a protein, and so it's got a gene, and it's actually a gene family. And so when people talk about actin, a lot of times they refer to muscle actin, and the same is true for myosin. A lot of times people refer to muscle myosin because actin and myosin in muscles is one of the other gene family members. This version of actin and myosin that are expressed in every cell type. Every cell type has actin and myosin, and it's using it that way. But the thing to remember, that's why I like to call it actin microfilaments, because it probably triggers your awareness of Oh yeah, that's involved in muscles and movement, because the actin microfilaments in the cytoskeleton play a role like muscle does. They play a mechanical role to pull things around. And so really good examples of that are shown in our book, just to introduce it. And there's three good examples. So the first one we already talked a little bit about, that the cell can change shape. It could be like a cube and it becomes more like a trapezoid. And like squeezing together, they always pull things together. And this is another really good one. So every time a cell divides, it divides in two, because a little string lasso grabs around the middle of the cell and contracts. And that contractile ring is actin microfilaments, and myosin is sliding them past each other to tighten it. If you had a balloon, you'd tie the string on the outside and tighten it, and you'd squeeze it apart to have two balloons. Well, this is like that, only the actin microfilaments are on the inside of the cell, or just on the inside of the balloon, and they're pulling it down like that. So two really good examples, and what's happening there, the cell is changing shape, parts of the cell are contracting. And then there's this other one, and this is one that's used a lot in the neural crest cells, and we just have to talk about it a little bit. I'll just show you a micrograph from our textbook, where if you just take any cell and you take it out of the body, you put it in a dish, and you just watch that living cell, the cell is round, or flat, but it's, got a shape to it, and it's just randomly poking parts of the cell out. And so let me draw that. Oops. And it's shown there, and so I'm sorry. And so let me just draw that out. And what it looks like is this: that we start out with just a regular cell, and it's got actin microfilaments in there, and they're polymerizing and depolymerizing, so they're just scattered around. And as it's doing that, it's polymerizing and depolymerizing pretty fast, and it's doing that, and it's doing it out here too. And sometimes it does it where they're kind of organized a little bit, and they go poking out like this. So we get a lot of actin microfilaments poking out there because they're polymerizing. And because of that, it's really important to appreciate that they actually are pushing the cell membrane out. They're still in the cytoskeleton, but the cell is pushing out kind of a finger. And you can imagine doing that, like pushing out inside a balloon. And you make a shape, as your finger pushes out, your finger is that microfilaments, like that. So the push out is made by these polymerizing microfilaments, actin microfilaments. They're not really using the myosin. They're using that polymerization energy to just push it out. And what's going to be key for us is when they do that, they're actually going to be pushing out these cell adhesion molecules, because the cell adhesion molecules are scattered all over the cell. And so anytime you push out any part of the cell membrane, you could you're pushing out these cell adhesion molecules. And the cell adhesion molecules might reach out and contact another cell adhesion molecule on another cell, and they might hold on. Or they might, we'll learn, and we haven't really learned about this yet, they might reach out and they're supposed to bind to some components of the extracellular matrix. So the targets of cell adhesion molecules are other cell adhesion molecules on other cells, or molecules in the extracellular matrix. And this thing that's poking out has a name, and it's called a filopodium. And so that's what that is, a filopodium. And the plural of that is filopodia. So it's just a little cellular structure composed of actin microfilaments, and they're pushing out the cell membrane and significantly pushing out these cell adhesion molecules. Just like you're reaching out your hand, you're climbing a rock face, you're reaching out your hand to try to grab the rock, another place you can hold on. So grab and hold on. And they do this pretty fast because the lifespan of one of these is just like a minute. or 2, 1 to 2 minutes. And the next thing to say about them is they're poking those out, those Philopodia, in most of the time, they're just poking out literally randomly from the cell. And so they poke out because of polymerization, and then pretty quickly they collapse back into the cell because of depolymerization. So they're growing, and then there's that image that it's a pretty good representation of what a cell looks like in a dish. They just have these Philopodia sticking out all over the place, okay? And that's just part of, kind of being alive, that animal cells are just doing that, kind of poking out and then collapsing back. So just to sort of draw that idea, we want to add now 2 things to that idea. And the one is, you can show this cell, and it's got, this one's growing out, and then here's one that's just growing out, and here's one that's collapsing back. And so they collapse back because of depolymerization. And that depolymerization occurs because they failed to have cell adhesions bind to a target. Cell adhesion molecules didn't bind to a target, a handhold, either in the extracellular matrix or other cell surface. Failed to have a cell adhesion molecule. Let's see, depolymerization failed because cell adhesion molecules didn't bind to anything. And I could say didn't bump into a target. So they just didn't bind it, so the thing just collapses back. So that's why they're short-lived, is when they don't find a target. If they find a target, then they hold on and it doesn't collapse. So that's one idea, is they're dynamic, they're just going to collapse again unless they bind to a target. Then they last a long time. The next thing is that sort of the second idea is they start out usually randomly poking out of the cell. Unless, it's a new term for us, but it's in the video for the neural cells, unless chemotaxis is happening. Chemotaxis is occurring. So again, you see them poking out, collapsing, unless they bind to a target. And they're poking out randomly, unless there's chemotaxis occurring. And if there's chemotaxis, it's not random, it's more directed. So we'll just kind of draw a figure like this and say, in a situation where there's no chemotaxis versus when there is chemotaxis, the cell looks different with regard to its Philopodia. And the difference looks like this, where with no chemotaxis, we're just poking out things randomly from the cell. I don't know, it's not a very good cell. It's starting to look like a dog. Okay, random Philopodia. And on the other hand, here's a cell where the Philopodia are coming out this way. And this is because of chemotaxis. And chemotaxis provides a way to orient the cell. And the chemotaxis means moving in response to chemical signals. And so this cell is going to start moving. In this direction it's going to migrate. And it's moving in that direction. It's getting orientation because of chemotaxis. There are 2 kinds of chemical signals. So chemical signals that guide migration come in two forms. One are called short range, and those involve direct contact between the cell adhesion molecules, and other cells or the extracellular matrix. So it's chemicals are guiding movement, and so that adds this idea that we're used to cell adhesion molecules grabbing hold, and maybe it's kind of a structural thing, or building a team, hang in there, let's hold together. Now we add to this that there's also going to be information. So when the filopodia reach out and make contact, there's going to be signals coming into the cell, communicating to the cell that says that's the way to go. We made some connections. We're following a breadcrumb trail, a chemical trail in the extracellular matrix, and we found some breadcrumbs. So back in the cell, there is some information input, biochemical information input, to tell the cell to orient its actin microfilament cytoskeleton to form filopodia in that direction. So those are short-range cues, and then the other cue, a category, is called long-range cues. And so long-range cues, short-range cues, or signals like that. And all these long-range cues are all secreted signaling factors, like the kind we know about. And I'll just redraw this messy diagram to get that image in there. And so when we say that actin microfilaments are involved in chemotaxis, it's because we're going to get oriented filopodia production, and it's going to be oriented either because of direct contact, short-range cues, between the cell adhesion molecules and the other cells or extracellular matrix material, or it's some other cell, and it must be a source cell that's releasing a signal. So this is back to the wind drawing assignment, where we have a source cell that's releasing a signaling factor. And so there must be a high concentration of those here and less of them here. And the migrating cell, the responding cell, has the receptor and responds, so it's a target cell. And in some cases, the target cell responds by moving toward the source of the signaling factor, or in other cases, it could be moving away. So it's either repellent, the signaling factor is either a repellent or an attractant. Okay, so that's happening, and it's talked about a lot in our video homework assignment. And let me just find that image to scroll to that. It will be somewhere here, sorry. If it's down here too far, yeah. So I sort of messed up, but I got that in there. But this is just a screenshot for your assignment, NeuroCrest assignment. And in red, that's going to be, we have a lot of stories in the assignment about the cytoskeleton and the cells changing shape because it's moving. And then we have some stories about cell adhesion molecules, and we have some stories about the extracellular matrix. Great. So the NeuroCrest drawing assignment is a great example of all the three things we're going to learn about. And in addition, it just briefly introduces this idea that the cells are migrating in a directed way. They're not making filopodia randomly. They're all moving that way because they're getting information put, and it's either direct contact through surface molecules down onto this cell adhesion molecules on the surface of the cell that's moving, or it's signaling factors, and the signaling factors are either say come this way or go that way. Yep. So we'll see examples of both those types of ideas. And so that's that image. And then the next thing to say is that when a cell starts doing that, it starts making so many filopodia. So it's just going to make a bunch of filopodia, and it makes so many, it just makes a little like edge of the cell, almost like like the edge of like the end of your shirt sleeve, where it just got a little piece of like almost like fabric, like a little ruffle coming out from the cell. And so yeah, these are filopodia, but this cell is got so much input for it to move in this direction that these all just get kind of merged into a little ruffled sheet, and that has a different term. It's called lamellipodium, or lamellipodia. So it's just a lamellipodium in the singular, and it's just lots and lots of filopodia. And so that's what a neural crest cell is going to have when it's migrating, because it's really moving in a directed way. And it's directed because of that chemotaxis story. And they're going to migrate. They're going to move in a directed way. And every cell that moves by chemotaxis is moving. Every animal cell that moves by chemotaxis is using that same mechanism that we see in our neural crest video. Okay, so that's a really good example. Now I got to scroll back up here. Really good example of still more of the microfilaments. So changing the cell shape to reach out with filopodia, or oriented way, lamellipodia. So that's shown there with the red in the microfilaments. All right. The thing our book doesn't show that's described, and I described it also last time, in migration, is that it's not, the force for movement is not produced up here. It really looks like it's going to be, right? Because we talked about, oh yeah, actin microfilaments have myosin motors, and the myosin motors cause them to slide by. So it really looks like a filopodia would reach out, make contact, and then contract and pull the cell forward. And what's not, I don't think described in our book at all, but it's just really satisfying, and it's described in our video assignment, is that the other example that we really like is that there's also actin microfilaments here. So again, those pieces of uncooked spaghetti just scattered here. And that's actually where the myosin motor plays a big role. And it's like when you squeeze toothpaste tubes from the base of the toothpaste tube, and it comes out the other end. We're actually squeezing the cell forward. So we reach out, make connection, and say, good, we're made connection, and then we push from the back to push it forward by squeezing, by having that contraction there. So that's a good image of sort of the three stories of changing cell shape that we want to learn about with actin microfilaments. And so that's a good introduction to the actin microfilaments, and then we want to talk about the microtubules, okay? So there is, in the assignment, a really good example of the microtubules. And so the microtubules are involved in moving stuff around inside of a cell. And so inside the cell, the microtubules look like polymers that make long, and I always draw them like train tracks, because they're really a track that a motor is going to bind to, and it's going to move. So there's going to be an ATP dependent motor that binds to those microtubules. And the cell can polymerize to build microtubules or depolymerize them, so it can build railroad tracks or break them down, and then these motors bind to them. And what's happening in the video that you'll learn about in the video is that these cell adhesion molecules are reaching out up here and holding on, and then the cell moves, and it moves over them. So back here, we need to be bringing the cell adhesion molecules into the cell and recycling them. So inside the cell, it's showing some little vesicles, and the vesicles are moving from the back of the cell to the front. And they're moving on ATP-dependent motors. So what's not shown in this image, and it's not, and it's talked about in our video, is there are these tracks like that in the microtubules that are being assembled and deassembled, and that allows molecules to be pulled down, of different types of molecules to get pulled down. So let's just look at that as a general idea here. I talk about a couple of examples, and so we saw that Drosophila nucleus in an egg that's maturing in the ovary, that at some point it is moved. So it's going to move randomly, remember, the microtubules are set up like that, so those are all microtubules. They're going to move randomly to a new location, and it moves because these motors attach to it, and these motors pull it to a new location. And they use ATP to pull it along. And if you know, like on a railroad track, there were these little hand carts in the olden days. People would stand on them, and they were like, had a little teeter-totter on them, and they just move them like that. And that's what these little motors are like. It's just a little cart, and they're just pumping along with ATP-dependent hydrolysis to pull whatever cargo, and it's referred to as cargo, pull it along. So we have an example of that. Then in this neural crest cell migration, we also have an example. So here's the neural crest cell, and it's moving along in this direction. And in that case, we have some vesicles that are in here, and we need to move them up to the front. And so there's also a microtubule train track. And it's not just one long piece of spaghetti. There are different pieces that are all lined up. And so the motors have to go and grab one track and then grab a different track. But there are these same motors, and the motors will grab those vesicles, and they'll pull the vesicles up to the front of the cell. So it's an example of that. See, and that's something that's different than cell shape changing. We're not changing the cell shape, we're just moving stuff around inside the cell. And from IntraBio and from our course, we've also talked about how anytime a cell goes through mitosis, it's going to have chromosomes that are going to need to get pulled apart to pull toward the opposite ends of the cell, so we can divide into two cells. So those are going to be chromosomes. And there we have microtubules bound to those chromosomes, because they're going to be the mechanical force to pull the chromosomes apart. And so that's true in every cell division on a eukaryote, is that we have these microtubules playing this role, and that's part of what's referred to as the mitotic spindle. And so those are memorable examples of microtubules, because you can see something's getting moved inside the cell. We're not talking about the cell changing shape, we're just moving stuff around inside the cell. We could move the nucleus in a fruit fly egg cell. We could move these vesicles in a neural crest cell in any vertebrate, or we could move chromosomes anytime a cell goes through mitosis. So those are good examples and memorable examples of, again, examples of stuff getting moved around inside a cell. Now we're just going to look at this different example and talk about that. And it's not a course in neurobiology, but there's another example that's very memorable in neurobiology that we can look at. And in our body, we have our muscles, and when our muscles contract, that's because there's some nerve that's stimulating to contract. And what that looks like is an example from neurobiology. And again, just a brief example to help set the stage. And in that example, here's the cell that's a nerve cell, and nerve cells are called neurons. And this neuron is going to make your big toe muscle contract. So it's going to be connected to your big toe muscle. And in vertebrates like us, all of those neurons that cause muscles to contract They're all inside the spine. And we have a special word for causing muscles to contract. They're called motor neurons. And so during development, what happens is this cell with its nucleus and all its ribosomes and stuff, it stays there, and it has to make a connection to the big toe muscle, and it does that by making a long connection that's called the axon, and this is part of the cell. So the thing I'm drawing there is the cell membrane. And then it's going to make a connection to the muscle. It's going to be right next to the muscle. And when the muscle contracts, it's because the neuron releases neurotransmitters. And this is a good example in muscle. It's also true in our brain. When we're thinking, it's because neurons are interacting with each other by releasing these neurotransmitters. The neurotransmitters get synthesized here, and they get recycled, but the cell has to be building them and sending them down here slowly and steadily because they get broken down and stuff like that. So the neurotransmitters, they get recycled and reused down here, but we got to get them down there when we make them. And so all along this axon is a series of microtubule train tracks. And we are pulling these vesicles along all the way, and they're full of these neurotransmitters and other things that the cell needs way down there. And so that's happening right now from your spine down to your big toe and all your other muscles. And again, the neurotransmitters get recycled, so they don't need to move these down there very fast. They're just slowly moving neurotransmitters because the cell is really long. And you can see this is a good example because it's true, but also there's a motor that's pulling them this way. And this is going away from the cell like that. And this motor in all animals that have nerves, this motor is called kinesin. And it's a microtubule motor that uses cargo. And so that was the first one that was discovered because our brain must just be full of kinesin. Because not just getting the neurotransmitters down to our big toe, but all of our neurons within our brain are communicating by neurotransmitters, and they have these connections, these axons. And so there just must be lots and lots of these motors. Again, you don't need them to think really fast, but you need them to kind of steadily resupply the end of the neuron with these. And so that's just a famous motor neuron because it's just so easy to think, yeah, there must be a lot of that in the brain, and it's pretty discovered. All right? So then we have that example, and now we're going to just draw this general drawing of a cell where it's going to be a cell, and it's going to just be a cell, and we'll make it look like it's kind of stretched out like this. And so there's the cell, there's the nucleus. And like we described, when you have microtubules and they are moving stuff away from the center of the cell, the motor that's always involved in that is out.
Well,
yeah, no, I remember there's a way to reverse that, but I can't remember how to do it all the time. Maybe just press it again? No, it's not that easy. Maybe go to white. No, it's not easy. Shoot. Yeah.
Well, we're in hell now.
Yeah. Yep. And
Have you tried the undo button in the bottom right?
just that.
Just hit that a few times.
Hit that, yeah. No.
Oh, no.
It's just okay.
Just the action. Yeah. Okay. So super. And there it is.
Oh,
Okay. So back to work. And So we had a drawing of a cell, and it's just a general drawing to get this idea that when the cargo is moving away from the center of the cell, that's going to be the kinesin motor moving it typically. It's kind of a detail, but just to see the introduction of this. So kinesin motor is using the microtubules and moving it away from the cell. And so then, not surprisingly, there was another motor discovered, and there are orthologs of these in all eukaryotes. And that motor is going to be pulling things toward the center of the cell, and that category of motor is called dynein. And so it's fascinating to see that, because in our neural crest cells, in that assignment, in the neural crest cells, we have a situation where we want to recycle the cell adhesion molecules. We want to get them from back here, the back of the cell, and we want to move them all the way to the front. So in that neural crest cell video, I introduced that actually both motors are involved, because we first start with the dynein motors to move that cargo up to the center of the cell, and then it has to shift over to microtubules with kinesin motors to pull it all the way to the front. So all that's saying is it's a good assignment for us to get good examples when we look at the migration of the neural crest cell, because there's the location where the kinesin motors are up in the front, moving it that way, so moving a lot of cargo, and that's where the dynein are located. And it's an ongoing process, and you know, you've seen, right, like a bulldozer, and a bulldozer moves forward on tracks. So the tracks are just going around and round and round. And that's also how a treadmill is built, right? So a treadmill has a belt, and it's just going around. It's the same belt going around and around. And so there's this really interesting process in neural crest cell migration and any eukaryotic cell migration where there's a treadmilling, and it's actually biologically called treadmilling. And it's where the cell adhesion molecules get hold of something, and then they just hold on, and the cell pushes itself up past them. And now that same cell adhesion molecule is back. We got to pull it inside and pull it on these microtubule train tracks to bring it back to the front. So it's very much like the treadmilling you see in like a bulldozer, where if you just painted one of the treads, you'd see it going around and around. But in this case, what it is not the tread. If we just tag one of the cell adhesion molecules, you see it going around and around. But the way it gets around, it has to go through this cycle. So it's just a really memorable example of microtubules doing their job. And they're just using it constantly, right? And it's ATP dependent. And so there is ATP dependent movement of the cell by the actin microfilaments pushing it forward. So that takes energy. There's also this ATP-dependent recycling of the cell adhesion molecules to keep constant reaching out. So it's just a really beautiful story of cell biology. And so that's a good example for us for that.
What's the green on the bottom?
The green on the bottom of this drawing is not from the book, it's not from our book, and the green is extracellular matrix.
Okay.
Yep. Okay. So good examples of what microtubules will do for the cell, right? We have 4 examples of movement of the Drosophila egg nucleus. I don't even remember what the other one was, but cell division, and also this Philopodia example and movement of these inside a cell. And I think there's one other one. Okay. And so here is that microtubule example that we've talked about before, about, oh, well, you need the mitotic spindle and the microtubules in it to pull the chromosomes apart. And this is the book showing that these centrioles or centrosomes get positioned, and it matters. And we learned that in plants, and we'll learn that in animals. That you learned in introbio, yeah, cell division occurs, great, it's just done. But as soon as the cell is in a neighborhood, like a layer of cells, you have this opportunity to either divide within the layer or divide between layers. And so that becomes really significant. And we have we'll have 3 examples of when orientation of cell division matters. There are many, many, many examples, but ours are, we already learned that it matters in a plant meristem microenvironment. Okay, so let's just look at that first example, and that looks like this corn. So this is going to matter with these layers on which orientation the cell divides. So if the centrioles and centrosomes pull the cell apart like that, great. If you pull it like that, it has different meaning. Right? So the same is true with animals. And in animals, we're going to use the neural tube as an example. And so this would be an example of where we were comparing plants and animals, and we said they have stem cells. That's analogous between plants and animals. They have stem cell microenvironments. That's analogous between plants and animals. And the stem cells can divide symmetrically or asymmetrically depending on the orientation of the division. That's analogous between plants and animals. So it's a really interesting phenomenon on our planet. And so what this is just showing is to remind you the neural tube is going to form. And when we get a neural tube formed, it's like a garden hose. The inside is hollow. The inside is hollow. And so this is liquid. And we have that in the neural tube, and it goes on for our spine and brain. This is where in our adult bodies, the cerebral spinal fluid is present. There's cerebral spinal fluid in the hollow center of our spine and also in the hollow ventricles in our brain. So that's liquid. So these are cells. And you can see these cells have an organization. There are more cells around them here, like that. So these cells have 2 surfaces. One surface is next to liquid, the other surface is next to other cells. And there are general terms for that. Maybe I'll just draw them over here where there's color. But to get this figure, you see, we get this setup where the cells that line the inner part of the neural tube have this sidedness to them. And let me just get it over here so there's some color. Okay, and it looks like this, where we'd have a neural tube, and this would be a cell, then we'll just draw one cell. So there's a cell with its nucleus. There's one cell, and see liquid, and this is other cells. So lots of other cells like that, okay? So the general terms are that this is the surface of the cell. That's called the apical surface, because it's next to liquid. And this is the side that's called the basal surface. So this basal surface. So this cell has an orientation just in where the cell is. It's in a layer, and it has an apical and basal surface. And there are some different proteins. So in the apical surface, there are proteins that are there because they're apical proteins. And the basal surface, they're basal proteins that are some different. Lots are the same, but there's some differences like that. Okay, so now in this layer, see, and it's a tube, so you know, it's a little bit hard to think of it as a layer, but it's a layer. They're in a layer of cells, and this cell could divide like that, and that would be a symmetric cell division, and we'd get 2 cells. And what these are in the neural tube is they're the stem cells. So the stem cells in our neural tube are in that inner lining that inner part of the neural tube. And I will just draw over here just a little kind of image of that. So like in a human fetus, if we did a cross-section of their brain, the cross-section of the brain would look kind of like this. You know, it's 2 hemispheres kind of joined together a little bit, and so this is just like a fetal brain. And in there are these hollow areas, and they're called ventricles. Pretty small areas in the brain, but they look like that, and they're hollow. And so the stem cells, let's give them a different color. The stem cells are the lining. That's where the stem cells are. That's where the brain is growing in a fetus. Those are the stem cells. And so we just zoomed on them, and we can have them divide, and if they divide within that layer, there's symmetric divisions, and we're increasing the number of stem cells. If we look at the neural tube in a vertebrate, there's lots of these symmetric divisions up in the anterior end, because we need more stem cells there, because that's where the brain is going to form. We need lots of stem cells up there, and so there's lots of symmetric divisions up there, and less symmetric divisions down in the spine, because we just don't need as many neurons along the length of the spine. So they could divide in that orientation, and then, you know, they could divide in perpendicular that. So we could also have them divide and create two cells. If they do that, this is the stem cell, and that's the precursor cell that's going to grow on to be a brain cell or a spine cell. So that's our example from animals to draw that similarity, that analogous similarity to plants. Where plants, stem cells are often in a layer, like a layer of cells. As they divide within the layer, they're stem cells, 2 of them. And if they divide between the layers, they're stem cell 1 and precursor 1. Okay? So that's why that example comes up. That's our animal example. And this just shows this from our book, but it's not until chapter 11, so not something we need to look at. But this just shows the idea that when the cell divides, well, it's got special proteins on the apical and basal surfaces. And if it divides symmetrically, both cells get the same proteins. They get the apical proteins and the basal proteins. But if it divides in the other orientation, one of the cells gets all the apical proteins and one of them gets all the basal proteins. And that's actually the difference. When the cells are in a layer, they have different surfaces. And we can either divide them so they have equivalent surfaces, some apical, goes to each cell, some basal each cell, or we can divide perpendicular, and the stem cell gets to keep all the apical proteins, and the precursor gets all of the basal proteins. So just a detail I had, just, and I don't know if we were going to get that chapter in our book, but just to show you that animal stem cell example. And the one we'll use is stem cell in neural tube. This is a layer. Then our third one is not covered in our book, and it's just a brief fact in development that we'll just point out as an example where the orientation of cell division matters. And where it matters is after egg fertilization and the early zygote, the early embryo, starts to divide, it matters how it's dividing. And so here's just a figure that you might not be able to see very well because it's from a different book. But what it shows here is a timeline starting with fertilized egg. And it's showing that if you look at all kinds of different animal embryos, the early embryo cells divide in specific patterns. So I'll just show, if we zoom in to one of the rows, it divides like this. You know, it divides into 2 and into 4, and then it divides in that orientation to get 8. And we saw a little bit, skips that a little bit, because that's how that early frog embryo divides when it's going through that really rapid mitotic cleavage. And that's called radial cell division or embryogenesis, and all amphibians do that. And so if we just look at the whole thing again, I'll just read off some of these names because wherever these are. So it shows this is a pattern of cell division, and all amphibians divide like that, and no other animal cells divide like that at the start of embryogenesis. Scientists saw this 200 years ago when they looked under microscopes, like that Carl Ernst von Baer, long, long ago. And they saw these patterns, and they were just describing embryos, so they just described hundreds of different animals, and they said, it's really curious that all amphibians divide like this, and no other animals do. They didn't have the feeling, that much of a strong feeling for the relationship, because this was before Charles Darwin and theory of evolution, and really that way that animals could be categorized. And so there are different ones, and so just to go down this shows, you know, this is a cell pattern that's only seen in echinoderms. This is in a variety of invertebrate worms. This is tunicates, here are mammals, here are amphibians, here are cephalopod mollusks, here's fish, here's insects. So it was a real surprise to those old-time people 200 years ago to see them all the same. Why wouldn't there be an exception? Today, we can appreciate, well, those are related. All amphibia are related, they have a common ancestor. The common ancestor must started its development by having those patterns of cell division. And then you just can't mess around with that. If you do, you know, your development would be defective. Why? Well, we can go back to the amphibians, because we remember that mom has loaded maternal factors in to different places. And so that mitotic cleavage is taking the fertilized egg and it's dividing the cytoplasm up like you're cutting a pie. And in order to get the proper pattern of cells with some factors from the vegetal pole or not, like that, you have to cut it out by having this controlled cell division. So it's just an example to add to our list of examples of how the orientation of mitotic division matters. It matters in the early embryo. They're just not dividing randomly, they're dividing with a pattern that's very much the same each time. And this is just the other one is just zoomed out just to show you, which is that's clearly a different pattern. And the thing that's different about those patterns is the orientation of the cell divisions, the orientation of the way the chromosomes are pulled apart, okay? So that's some stuff about cytoskeleton. Now we're just, and so, you know, some background, add a little bit of examples, but we'll add some more examples, like when you do your neural crest cell migration video, you'll be like, okay, there's roles for the actin microfilaments, there's roles for the microtubules, and that's what we want to take from that example, and we'll have a couple of other examples from the book. The next thing we want to do is introduce the cell adhesion molecules a little bit. So we've done that already a little bit, but it's just a minimal intro, like, you know, cell adhesion molecules are like the variety of macaroni, okay? But we know it's important because we saw that example where you could remove all the cells from an organ, and that would allow the extracellular matrix to remain. Oh, sorry, that's kind of out of place. We'll get to that when we talk about the extracellular matrix. Here's the examples of molecules that we should learn about. Oh, I jumped here too. I'm out of order. Let me go to, where did I have? Is it still coming up? Okay, let's go to cell adhesion molecules. And for that, our story is entirely, to introduce it entirely in just one page of the book. So for us to learn about cell adhesion molecules, just go read that page. There are lots of different types of cell adhesion molecules, hundreds of different cell adhesion molecules in our bodies. So how to introduce it? And the book decided to just choose 3 examples. And so we're going to learn examples of 3 big categories of cell adhesion molecules. And it's 3 different families of proteins. And they have a lot of different roles, but this is just a one-page intro to cell adhesion molecules. So the book says, let's pick 3 and learn 3 examples in order to introduce some highlights, okay? And so here's the 3 examples. They're figured on here, and they're showing kind of nicely in this that there are 3. There are 4 panels because the first example is got 2 of the panels. So there's 1, 2, 3. And the protein families are called cadherins and cams and integrins. And those are all cell adhesion molecules. And they're all proteins, and they're all members of families of proteins. Yeah? And so the book shows a figure because they're all on the cell membrane, so the gray thing is the cell membrane, and there are two cells that could interact. The first two, cadherins and cams, are like the macaroni we learned about in the assignment, where those cell adhesion molecules bind other cell adhesion molecules on another cell. So here are two cells, and we've got cadherin on this one and cadherin on that one. So they're gonna hold hands. We've got this cam on this one and a cam on that one. We're gonna hold hands. Then the integrins, you can see they're not holding hands between cells. So the reason the integrins are used in this example, because integrins like to bind to components of the extracellular matrix. So, right, that's something we already mentioned, and we want to emphasize that, is that cell adhesion molecules, yes, they can hold on to other cells by holding on to cell adhesion molecules. That's what cadherins and cams do. Or cell adhesion molecules can hold on to part of the extracellular matrix. That's what integrins do. So that's an example of how the book says, let's take a really complicated story with lots and lots of players, and we'll pick three famous ones, and we'll pick them because they're famous, they have a lot of roles in our body and health and disease, and also because they'll teach us some satisfying principles. And one is, well, they could bind to other cell adhesion molecules on other cells, or they could bind to the extracellular matrix. Okay? Then the next reason why I pick cadherins is because cadherins hold hands, they hold hands with other cadherins, and they hold hands in a calcium-dependent way. So that's nice because it gives you the idea of we can hold hands and we can let go. And all we have to do is hold hands by keeping the calcium concentration high, and if we lower the calcium concentration, we'll let go. So that's nice because it adds this idea that these can be dynamic. And they're all dynamic. It's just like we want to try to find some memorable examples where you're like, yes, they're dynamic. Okay, so that's why cadherin comes up. And you can see cadherin is kind of satisfying because it's got that Ca, that calcium, in the front of the word. Calcium cadherin, right? And so that's another reason, is we want to show that they can hold on or let go. And all these could let go, but just to kind of emphasize, yeah, there's cadherin is really good at letting go, and he's really used a lot in dynamic situations like cell migration. Let's pause, we'll do a concept chat. And maybe, yeah, we might not have any time to do anything more, but we'll do a concept chat. So find a partner, and you know, if there's somebody you haven't sat down with in class this semester, you can say hi to them. And we'll quickly talk about three important things.
Especially like calcium, that's how it pulls let's go. So kind of want to use like
Yeah, it's funny. So I did my bio so I know he's very big. I think I heard kind of general, but I don't. That's awesome.
There was a lot. I like how he gave us 4 examples for
It's always fun.
So we have the movement of the fruit fly, cell division, migration of the cell, and
Yeah, honestly,
it's just the receptors.
I think it just threw a series of 6, like the 16 sigma.
Like, what do you use the receptors?
Yeah, so it's like constant learning. So it's like one piece at a time. Or like for like a little
After
so
like
like
until like
one of them like this one right here, like literally like gonna go around. So as it gets tough, she just like continuously grow.
Yeah. Well, it's also like helping them.
Yeah, that's great.
That's like part of the stuff.
Just we'll do this for another minute.
Yeah. Yeah, and I kind of just like always like kind of going around. Yeah, and they're just they're always choosing that.
Okay, and so Brianna, what's one thing your group talked about?
We talked about the philopodia and how with polymerization of the microfilaments, it will randomly stick out unless it's happening during chemotaxis, and which needs like either direct contact or like a signaling factor.
Yep. Yeah, yeah, and every time I teach, I'm like, oh wow, that would just be really excellent as a series of tattoos, right? Huh? Super cool, like up, down your arm or your two arms. Like, check this out. Yeah, and but yes, that story of those cells reaching out with philopodia, and then when it gets really oriented and real strong chemotaxis, those philopodia are so crowded they become this sheet of lamellipodia. Jeffrey, what's one thing you guys talked about?
We just talked about the actin like either being random or directed via like chemotaxis.
Yeah.
And
Yeah, so that's about the idea that we can look at the location of actin in a cell, and the actin cytoskeleton in the cell, those microfilaments, look like long pieces of uncooked spaghetti, and most of the time, all the time, they look kind of like somebody just dropped them. They look pretty scattered, but they're being polymerized and depolymerized, and that organization can be shifted to be not just random scattered pieces of spaghetti, but start polymerizing it so it starts poking out more often in that direction. So we have more of that polymerization and more poking out, more philopodia in a directed way. And that's going to help the cell orient its migration or movement, because it's going to be oriented through chemotaxis signals, either direct contact between cell adhesion molecules and other cells or the intracellular matrix, or it's going to be getting some signaling factor signal where it's orienting that polymerization. That's what's going on to do that. And then one more from Naomi. Yep. Okay. Great. And so we have a couple more minutes. And do people have questions about anything at all right now? Yes, Alex.
Talking about the different sides of the cell, the apical side and the basal side, this is like just kind of like an offshoot of that. I was just curious when like there's like basal cell like carcinoma and stuff, does that mean that there's like, does that have anything to do with like the cells on the outside like that?
No.
No.
Yeah, no, because that word basal means a little bit like it's a little bit false, but like a little bit more like the ground, because it's kind of surrounded by other cells, so it's not next to the liquid.
Okay.
And a basal cell carcinoma, my understanding is it's part of that region of like the basal region, or more that chunk of cells that are next to that.
Okay.
So that's not an issue with that sidedness of a cell, but the kind of that area.
Okay.
So yeah.
Okay.
Yeah.
I just have a confirmation question. So inside the neural to asymmetric division.
Yeah, so let's just clarify that. And so I guess that's still that drawing. And so what I'm trying to draw there is that, and we can just redraw it, is you could say, here's a cross section of a neural tube. So this is a neural tube, and And that's liquid center. And the tube is long. It goes from posterior to anterior. And it's eventually going to look like this. The central nervous system is going to have a hollow center in the spine. So that's going to be a hollow center in the spine. And then it's going to, in the brain, those hollow regions are going to be called ventricles. So a little different structure, but the spine, just to now draw the spine, that's where the cells are. The brain, that's where the cells are. And we have this hollow center. And you said, in our body, in any vertebrate, where are the cells that are making the brain? Where are the cells that are making the cells? Where are the cells that make all the brain cells and all the spine cells? And the answer is, they're just right here. They're all along the lining of the liquid part. And you call it a membrane. I wouldn't call it a membrane. I just think those are the cells that are right on the surface. And because of that, those cells, those are the stem cells. Neuronal stem cells, they're all there. So if you looked in a fetus and you're like, where are the cells dividing? They're just dividing there. That's where all the growth occurs in the nervous system. And they're in a layer because they're in, just along there. So all the stem cells have apical and basal surface. And that's why we have a situation where they could either divide, and stay on that same plane. So now we're going to have them stay in the same spot, and we're just going to get more stem cells. And there's a lot of that happening up here because we need a lot of stem cells to make a lot of brain cells. You just have to increase the population of stem cells. And the alternative is they divide and they don't have the other cell stay on the same plane. It gets pushed out of that surface area, and that's a precursor cell. Yep. Other questions people have? All right, so we have to stop. There's a couple emails I still have to catch up with on people on grading, and also comments about your term papers. With your term papers, I can make some comments by email, but super helpful to just talk briefly in person. And you can hand in your flowering assignment. Is that due today? No. Is it due today?
It's due today.
Yeah, the flowering assignment is due today. So if you're not handing it in, you should email it to me. I've gotten several by email already.
I hope it's due today.
I think it is. Let's say it's due today. And